We have already discussed about the importance of signal peptide. Actually, they are important for the translocation of the proteins like secretory, and those are seen on plasma membrane, especially transmembrane proteins and lysosomal proteins, etc. These signal peptides are seen in the N-terminal region of a protein. These signal peptides are synthesized at first during the translation process, and these are the first regions of a protein that are exposed to the cytoplasm. Once it gets exposed to the cytoplasm, then these may be recognized by specific proteins known as SRP. SRP means they are signal recognition proteins. So these signal recognition proteins, or SRP, they recognizes these signal sequences or signal peptide. Along with the signal sequence, it also interacts with the ribosome. So at a time it binds with ribosome as well as signal sequence. Hence, the further translation process gets blocked. So this SRP that interacts with the signal peptide and ribosome, as a result of it, the translational process get inhibited. Then the translation can resume only when the entire unit that may be translocated on the surface of endoplasmic reticulum. On the surface of the endoplasmic reticulum, there are two kinds of receptors. They are translocon. That translocon can interact with the ribosome, and the second thing is SRP receptor. This SRP receptor that can interact with SRP, and also it can exist in two forms. They are SRP GTP bonded form and GDP bonded form. So once this unit interacts with the translocon, then the SRP that interacts with the SRP receptor. See, normally the SRP receptor that can be found as GTP bounded form. GTP bounded form. This binding, that means SRP receptor and SRP, that activates the hydrolysis of GTP that is present in the SRP receptor. So GTP gets hydrolyzed and it may be converted to GDP form. So now the receptor is SRP receptor GDP bounded form. Now it having a different conformation, so now it cannot interact with SRP. So this SRP may get eliminated. So the inhibition for translation gets lifted. As a result of that, the signal peptide gets free, and that signal peptide goes to the luminal side of the translocon. Now the hydrophobic region or stretch of that signal peptide interacts with the hydrophobic region of the translocon. The energy released from the SRP receptor that may be utilized to widen the translocon. So now the translocon gets widened, then the translation process gets continuous, and the polypeptide that is synthesized in the ribosome that may be translocated into the luminal side of the endoplasmic reticulum. This is the way in which the secretory proteins are synthesized in rough endoplasmic reticulum. And talking about the transmembrane protein, the mechanism is almost similar. They are also having an N-terminal signal peptide or signal sequence. In addition to that signal peptide, they are having an internal structure of hydrophobic region. It's having or maybe composed of around 20 amino acid residues. Once translation process gets continuous, then the N-terminal signal sequence along with the growing polypeptide chain that may be moved to the luminal side of the endoplasmic reticulum. See, N-terminal and the growing polypeptide chain, they are now in the luminal side of the endoplasmic reticulum. See, this is the luminal side, this is the endoplasmic reticular membrane, this is the cytoplasmic side. Okay. Once the transmembrane sequence or the stop transfer sequence gets synthesized, then that will be translocated into the translocon. The internal or the luminal side of the translocon is hydrophobic in nature, and now this hydrophobic stretch, transmembrane stretch, that is also hydrophobic in nature, so it can interact with the translocon, and the internal side of the plasma membrane, that is also hydrophobic in nature. That's why now this hydrophobic stretch can move to the plasma membrane. See, now this hydrophobic stretch that may be translocated into the plasma membrane, and then the further polypeptide chain continues. So eventually the translation process gets over and the entire polypeptide may get synthesized, and but this stop transfer sequence or hydrophobic transmembrane segment that really stopped the entire polypeptide to move inside the luminal. That's how it became transmembrane protein. Actually, this is a single span membrane protein. In the case of multi-spanning membrane proteins, they may be having multiple stop transfer sequences. So at each time the translocon gets experiences with the stop transfer sequences, it gets incorporated or moved into the uh, endoplasmic reticular membrane, and then the further regions of the polypeptide gets synthesized. Once the translation gets over, we can see that the polypeptide that may be anchored in the membrane with the multiple transmembrane segments. The orientations of N-terminal and C-terminal regions are dependent on the number of stop transfer sequences or hydrophobic transmembrane segments.